Good afternoon, everyone. On behalf of the HNRCA, it is our pleasure to welcome Dr. Ann Newman. Dr. Newman is a professor and chair of the Department of Epidemiology at the University of Pittsburgh and is director of the Center for Aging and Population Health in the Graduate School of Public Health. Dr. Newman also has a joint appointment in the Division of Geriatric Medicine in the School of Medicine. She is internationally renowned for her work in the epidemiology of aging, longevity, and disability. Dr. Newman's research has defined the impact of apparently subclinical disease on physical, cognitive function, and on metrics of healthy aging. She conducts clinical trials to prevent disability using physical activity, weight management, and medical therapies. Dr. Newman has published over 800 research manuscripts in major journals and has been recognized among Thomson Reuters' most highly cited researchers. She served as a member of NIA's National Advisory Council on Aging and currently serves as a member of NIA's Board of Scientific Counselors. She's an associate editor of the Journal of Gerontology, Medical Sciences, and served as the editor-in-chief for the journal between 2016 and 2020. It is our pleasure to hear from you, Dr. Newman. A gentle reminder to our audience, please put in your questions in the Q&A box and not in the chat box, and we would be happy to have Dr. Newman answer them for you. Again, welcome, Dr. Newman, and take it away. Thank you.
Thank you very much for your kind introduction. It's a pleasure to be here and an honor to be invited to speak at your institution. I'm going to share my screen. Okay. So what I thought I would talk about today is about my perspective as an epidemiologist in the study of aging. Entitling my talk, Analyzing Aging, an Epidemiologic Perspective. And my goal here is to get us all thinking together about age and aging, perhaps in some new ways, to think about aging as a number and how that relates to age as a biologic construct. I think that everyone is very well aware that age is a strong risk factor for morbidity and mortality. This is an example of the probability of developing cancer within age intervals in the United States. And the increase in the risk of cancer is quite dramatic from middle age to up to age 70 and over. Again, this is very well appreciated. One of the questions is, how strong is age as a risk factor compared to other risk factors? You can't stop yourself from aging, but maybe there's other things that you can do. Here's an example of the risk of cardiovascular disease. This is the 10-year risk of cardiovascular disease in men and women, comparing non-smokers with no other risk factors to smokers, again, with no other risk factors. So just looking at smoking alone, you can see that the smokers have a higher risk of cardiovascular disease compared to the non-smokers. So that in people over age 70, particularly if you look at the blue bars in the men over age 70, the risk is far higher. But when you really look at age, this age gradient is sixfold. So smoking gives a twofold higher risk, but age over 20 years, the risk is sixfold. So it's a much stronger risk factor than something that we consider to be a very strong risk factor. Another thing in thinking about chronologic age is that age is not age, it can interact with other factors. This is the risk of breast cancer by the age of menopause in the Nurses' Health Study. And what's notable here is that for women who have an earlier menopause versus a later menopause, the risk can be much higher. So for those who have a later menopause after age 55, the risk of breast cancer actually diverges, and this is thought to be due to a longer exposure to the estrogen cycles. So age is not age is not age. It can interact with other factors. Here's an illustration of the risk of hip and bone loss with advancing age. These are longitudinal results from the study of osteoporotic fracture. And what's notable here that you might have noticed with the first slide I showed on cancer is that in the oldest group, the risk is much higher. You see kind of a stepwise increase in the risk with each age group, but then much, much higher in the older old. So that we need to think about the acceleration of risk in the oldest people age. Another thing that people often forget to think about is that some risks are age specific. This is data showing age-specific trends in colon cancer. This is the older population at the bottom, where the risk of colon cancer, in fact, has been going down, 85 plus, 80 to 84, so on and so forth. But then when you get to these youngest age groups, there has not been, the decline has not been realized in reducing the risk of colon cancer. And in fact, if you look at age specificity, the risk of colon cancer has actually gone up in the youngest, in the 20 to 29-year-old age group. And the point of this is that we often think about age as something we can simply adjust away. So if you want to know what the risk of colon cancer is over time and you age adjust the risk, you're going to see, since most of the cancers are in this oldest population, you're going to see that the age-adjusted risk goes down. But that would mask an age-specific risk going up. So again, caution with simply thinking that age can just be adjusted away. Here is another very profound example of this pattern where mortality rates in the US, age-adjusted mortality rates, have been going down and going down for all causes of death. But then when you stratify by age, this is age 45 to 54, it was recognized more than 5 years ago that there is an increase in, these are mostly deaths from opioid overdose. And if you looked at all causes of death, if you just age adjusted all causes of death, it would appear that that there's been stability or decline. Whereas when you age stratify risk, you begin to see that age is not age. And in the younger middle-aged population, the opioid epidemic was missed for quite a while because of simply age adjusting rates. So in analyzing age, you need to ask the question, is the pattern with age, is it a linear pattern? Is it accelerated? Is there a threshold after which the risk pattern changes? Interactions may exist. I showed you the example with hormone replacement therapy that age has a different meaning depending on, not hormone replacement therapy, but depending on the age at menopause. And then is there age specificity? Sometimes stratifying by age, and in studying aging, we're beginning to recognize that that the oldest need a special look in particular. So the question is age just equal to time? And I found this cute little picture of people sort of standing on their timeline of their lives on a measuring tape. Is that what you're really doing when you're adjusting for age, is adjusting for time? Here is an example of an analysis that we did some years ago in the cardiovascular health study, where we thought, well, if age is just time, if we could adjust for all risk factors and for all diseases that are present, and we had 78 factors that would account for these time-dependent exposures, maybe we could make age as a number go away. This is the risk of age after adjusting for everything that was age-related that we could think of. And you see the total mortality rate was still strongly related in a graded fashion to age for total mortality, for cardiovascular mortality, for stroke, for cancer, and most dramatically for dementia, where we couldn't even fit the whole line on the bar. For every cause of death, we could not explain age as a number or age as just simply the passage of time in mortality risk in the cardiovascular health study. So what does age tell us? Well, there's a lot of information besides time. It tells us about cohort effects. Exposures may differ depending on when you're born. Two cohort studies that I've worked on, health, aging, and body composition, versus the SOMA cohort, which we just finished recruiting for this month. In Health ABC, we studied people who were born between 1916 and 1925, and they were enrolled about 25 years ago. Well, those individuals went through the 1917-18 flu epidemic, the Great Depression, and grew up without any antibiotics, basically. There might have been one or two, but it was before penicillin. The SOMA cohort now, their birth range was 1936 to 1951, a very, very different era to be born into. Now with antibiotics, now with cardiovascular disease prevention, cancer screening. But on the downside, going through the 2008 recession and now the COVID pandemic. Age also probably doesn't adequately capture, even if you think of it as time, the time-dependent exposures and how they accumulate, environmental exposures I have listed here. It doesn't tell us what the presence of chronic disease is, and it doesn't tell us anything about the person's intrinsic capacity to repair. So there's a lot more information in there. I have a picture of a tree in cross-section in some early conversations that I had with some trainees. Some of them said, well, with a tree, you can just cut it and count the rings. Is there anything like that in people? Well, unfortunately, trees and people are very different and much more complicated. So how do we define aging? I define it as the accumulation of damage and repair that occurs in cells, tissues, organs, and at the whole person level, that occurs over time and that results ultimately in a loss of function and a vulnerability to disease and death. So far, though there are examples of altering aging, we can think of aging as being universal. Generally, it's detrimental and it's resulting in a loss of function and vulnerability, and so far inevitable. So now I want to turn to talking about analyzing aging versus measuring aging. There are several things that have been proposed to measure aging, and I'll address some of these. Aging indices of multiple factors, biomarkers of age, and aging clocks. And these are just some illustrations of those. Now, one of the things that I do is that I teach, we have a training grant in the epidemiology of aging for pre and postdocs. And one of the things that I do is to try to distinguish between disease and aging, which is really not really possible, but conceptually, I think it is. In this model, we think about modifiable risk factors that lead to cellular damage, repair, altered tissue function, which leads to altered organ function, disability, and a vulnerability to mortality. But we think about diseases as being selective and preventable. We think about the external, personal, environmental, and contextual factors that lead to diseases. Whereas with aging, we're talking about the intrinsic failure of a biologic system, but it runs through the same pattern of altered cellular tissue organ function. But the aging phenotype we think of as being universal and inevitable. In this model, modifiable risk factors can cause intrinsic factors to be damaged so that we can think about risk factors as accelerating aging. Here I've highlighted the disease pathway, modifiable risk factors leading to disease phenotypes. And for most of epidemiology, people have focused on this in terms of disease prevention and have not focused on aging, as this has been thought to be fixed and that you can't do anything about the intrinsic aging process. But geroscience now has shown us some of the links between the basic biology of aging and diseases and functional status. This is one of the illustrations from a report by Philippe Sierra on the NIA website. And I like this illustration because it links aging biology to aging physiology, which I think is really important in understanding how we can use aging biology to ultimately improve the aging process. So what are some potential biomarkers of aging processes? Well, you've probably heard of some of these and may have had lectures on any number of specific processes. I have here telomere length, advanced glycation end products, clotho, cystatin C, NT-proBNP, senescent tissue cells. But we've also looked at some of these person level things like lens transparency, arterial stiffness with pulse wave velocity, processing speed, maximal oxygen consumption, muscle strength, and gait speed. And I think it's useful to focus on these latter three as they're whole person measures that really can tell us something about the aging process that could possibly be intermediate outcomes in interventions. So we know from the Baltimore longitudinal study that VO2 declines with age. And I like this particular study because they looked at VO2 in people who were at various levels of physical activity throughout their lives. These are trajectories of a period of 10 years for each age group, and then they're kind of knitted together. And then the dotted and dashed lines distinguish those from at the highest level of activity from those at the lowest level of activity. So at every age, those who are more physically active, activity level 3, have the highest VO2. But as the different cohorts get progressively older, you see the same decline in VO2 regardless of the level of physical activity. So physical activity is shoving people up higher in their level of capacity, but it doesn't seem to be changing the trajectory of decline. We also see this for something like grip strength. These again are knitted cohorts combining harmonizing cohorts in the UK where grip strength was tested at many different ages. And again, you see that there is this decline with age in every study that has been done. Here's some work that we did in the cardiovascular health studies in a group we called the All-Stars, who were the longest term survivors. But we looked back at all of the data collected in CHS over about 25 years. And we graphed the change in gait speed. We started around age 65 in CHS, so I can't go back younger than that. We saw an initial little increase, which could have been a training effect. And what we did here was we separated those who were healthy in the dark lines from those who had chronic health conditions. And we analyzed the trajectories. And you see that at every age, those with no health conditions were had higher gait speeds, but regardless of being a healthy status or an unhealthy status, this decline was pretty similar. And I think of this really as these measures as helping us define the aging process, sort of intermediate measures that can help us to see how someone is changing over time. And we know that all three of those measures are strong predictors in older adults, strong predictors of mortality. But what we need now is measures that we can track people's health over time and try to improve the process. Another thing that we did in that particular analysis was we looked at the trajectories of function. We looked at digit symbol substitution test, a cognitive test. We looked at the mini mental state, another cognitive test, and gait speed and grip strength. And the function tests, you see all the function tests in men and women, regardless of their health status, you see the function tests going down. And this over time was correlated with increases in IL-6, cystatin C, and adiponectin. So telling us that perhaps we can map these blood markers as potential biomarkers of aging if we think of the decline in function as capturing aging. This idea of using multiple tests to try to capture the aging process is not new. This is a paper from 1980 from some work done in the 1970s in the Baltimore Longitudinal Study on Aging. Here they show an individual person and some of the things that got worse or got better over time for one specific individual. And they found that their lung function got worse, their bone definitely got worse, and some of their cognitive tests slowed down. There were, in this case, some improvement in creatinine clearance, but they proposed in the BLSA to take a profile like this and to use that to estimate a global measure of biologic age. So this is not a new idea. This goes back more than 40 years. Note that they also noted this heterogeneity, which I think is an important concept, is that aging does not necessarily go on in the same amount in the same direction in every system. So using that idea of using multiple parameters, there have been multiple efforts to define a biologic age using fairly routinely measured tests. This is one from Morgan Levine from now 10 years ago. You might know her name from doing a lot of work with epigenetics now, but basically found that these measures all contributed to giving a better estimate of someone's biologic age, in that a metric of an adjusted age, including chronologic age and all these other measures, could predict mortality more accurately than age alone. And subsequently linked this biologic or physiologic measure of age to epigenetic age, which I'll show you. There's also an important paper from Dan Belsky, who took a similar list of commonly measured tests in younger people aged 26 to 38 and was able to show not all of them got worse, but they were able to put together a biologic aging across multiple organ systems in middle-aged people, and considered this to be demonstrating their biologic aging. So one of the other things that we've done here at Pitt that I wanted to share is to really focus on altered organ function. And I've highlighted that in orange, again, showing you my same model of the modifiable risk factors leading to disease and the intrinsic factors leading to aging phenotypes. Here we use the concept of subclinical disease. Measurable disease is quite substantial, accumulates in people even if they haven't had a clinical health event. We've shown that there's a strong correlation between these measures of subclinical disease and frailty, as well as function. And it's capturing atherosclerotic burden, silent strokes, asymptomatic lung disease. We know that each of these individually has a strong relationship with walking endurance, cognitive decline, and ultimately mortality. So we put together this scale for these 5 measures that were available in the cardiovascular health study: the carotid wall thickness from ultrasound, the forced vital capacity from spirometry or pulmonary function testing, white matter grade from a brain MRI, serum fasting glucose from a glucose tolerance test, and serum cystatin C to assess renal function. And we graded these just on a 0 to 1 scale. And we did that partly because when you did it that way, you ended up with people having a 0 or the best, having values for these that would be considered physiologically normal for a young person. I would note that we found little clustering between systems. They seem to be contributing independently. And when we added them up, we were able to explain 40% of the mortality risk due to age. It actually was as good as chronologic age, but we wanted to come up with something that didn't have chronologic age in it. And so explaining 40% of the effective age without using chronologic age, we thought made the point that we were capturing proportion of the information that's buried in that number. We were particularly interested in finding the super healthy individuals, who are very hard to find when you just define people as healthy on their medical history. You wouldn't find the people who are actually super healthy for their age. We also extended this to a simpler phenotype using what we call a healthy aging index with more commonly available measures: systolic blood pressure, fasting glucose, forced vital capacity from spirometry, creatinine from a standard blood test, and a mini mental state exam, which is a lot easier to get than a brain MRI. And again, we were able to show this very nice graded relationship in the risk of mortality that explained not as much, but a substantial proportion of the age effect. So why do we care about this? Well, we now know that aging is modifiable. We have known for many years that caloric restriction modifies aging, that genetic manipulations, you can knock out or find mutant model systems with changes in the growth hormone axis and insulin signaling pathway. There are studies on stem cells that show that some of these giving people stem cells could potentially rejuvenate them, but it's not just cells. We know that there are proteins from parabiosis experiments where you tie the 2 animals together and make the old one makes the young mouse older. The young mouse can rejuvenate the older mouse. And these are thought to be mediated by signaling proteins. And then we know that removal of senescent cells in vivo can also rejuvenate an organism. So if aging is modifiable, then how are we going to measure it? Well, some of the reasons that we might want to do that is that it might be useful to have a better biomarker of aging or aging indices to predict the tolerance of a stressor. So you might want to get your biologic age checked before you have chemo or major surgery. It might be a target for prehab or for rehabilitation. It might be helpful in identifying intermediate markers. So are some of these biomarkers in the pathway to improve function, and can they be used as intermediate markers of benefit? Could they be used for prognosis, say for screening, or could they help us identify molecular targets for not only for diseases, but there are some aspects of aging that we don't really address that are problematic, such as lens stiffening, hearing loss, peripheral nerve impairment, vascular stiffening, loss of muscle strength and cognitive decline, and slow wound healing, that some of these markers might help us to begin to address some of those in a better way. So what would be some criteria for informative biomarkers? Well, we think that they should be representative of the biologic aging process. They should be known to change with age or to differ between younger and older. They should be known to predict mortality and functional decline. They should be reliable and feasible and stable over time, and preferably non-invasive. So blood or urine tests, perhaps saliva or hair samples, or imaging tests. And they should be fast and cheap to apply in research to larger populations so that they're feasible ultimately for clinical use. This is a very nice paper that Jamie Justice put together when we were developing a blood-based biomarkers proposal for the TAME trial of metformin, which has been not quite started yet because of the pandemic, but hopefully will be. And ultimately, it should be responsive to intervention. I'm going to skip that and go to this one. This was the framework in that paper for selecting biomarkers that had a strong association with age, that were validated in being also associated with physical and cognitive decline, and represented to some degree the biomarkers of the aging process. So one of these is DNA methylation. You've probably heard about methylation clocks, which are marking the processes of the control of DNA as it leads to differentiation and tissue homeostasis and ultimately aging. And this illustrates that some people can have methylation markers that would say that they're older, whereas others might have markers that would make them biologically younger. There are several approaches. The first Horvath one was optimized to chronologic age. The second one also to chronologic age, but limited to blood, whereas the first used tissue. Morgan Levine, who I showed you her biologic aging, she linked 513 CPGs, which are methylation sites, to phenotypic age, which I showed you earlier. And more recently, Lou showed the markers that were most strongly related to mortality. Interestingly, it was very strongly also related to smoking. Here's a visual representation of what these different CPG sites are related to. Again, you see here the Lou, called the Grim Age clock because it was linked to mortality, or the Grim Reaper, was picking up smoking, depression, poor lung function, low socioeconomic status, where the original Hannum clock was less strongly related to these external drivers of impairment. Unfortunately, we still don't know much, but we're learning a lot all the time about how methylation age is related to biologic function. It's important to note that it hasn't been related to senescence or to proliferation potential of cells. So much more work needs to be done on that. Another one is telomere length. These are the caps of the chromosomes that protect the genome from damage, and they shorten over time in somatic tissues. And there are sort of tissue-specific aging syndromes such as pulmonary fibrosis or bone marrow failure that are clearly related to telomere shortening. But the way it is measured in people, it's usually measured as an average when the cells in the blood have different telomere lengths, and cells in specific tissues may not be as highly correlated as one would like to the blood. Interestingly, longer telomere length is related to cancer. This is a meta-analysis showing the risk of cancer using a Mendelian randomization study of some of the genetic markers that relate to a longer telomere. So people with naturally longer telomeres actually appear to be at higher risk of cancer. And it may be that the price we pay to avoid cancer is being able to put the brakes on the telomere length. So it's not really clear what's optimal for telomere length. Another area in biology of aging is the senescence-associated secretory phenotype, or SASP, where damaged cells undergo senescence, meaning that they can no longer divide, and they produce cytokines and inflammatory markers. These are thought to lead to cancer. This is a very promising area because interventions have begun to be done, but it's very hard to measure. And it may be that a certain panel of cytokines may prove to be a useful marker of senescence. It may be that tissue is needed. Here's an example of a tissue study where senescence in fat cells was detected using this biomarker of P16, but it probably is not, there's not one biomarker that tells you for sure that a cell is senescent. And it's also not clear from this tissue study if those are fat cells or other cells in the tissue. Nevertheless, preliminary analysis showed that those with more senescent cells had worse function, which is very promising. So we have a lot of challenges with aging biomarkers. The telomere length, we have mixed populations of white cells, and it's highly variable. It hasn't turned out to be very useful in measuring change. The methylation clocks originally were tuned across the lifespan. They reflect, though, by tuning across the full lifespan, both the developmental program and aging. And I think that this is why it differs from those later methylation clocks that focus on mortality risk or a physiologic or biologic age phenotype with other measures. So I think the later methylation clocks are going to prove to be more useful as they're measuring that period of decline. And then these other biologic age markers that I talked about, they better reflect the period after development, but they could represent markers with a strong environmental influence, and we don't know the effect of birth cohort and peak development on some of these. This is a paper that Dan Belsky did looking at how these different clocks are correlated, and these different measures like telomere length or a biologic age, such as he and Morgan Levine did with common risk factors. And the correlations between all of these measures was not very strong. This group of measures at the bottom right were all based on those risk factors like your cholesterol and your VO2 max. These CPG clocks were across the developmental life course, so they might have been capturing development as well as decline. And they're kind of grouping together there. But between those two groups of types of measures of biologic age in here, they're not very highly correlated. Nevertheless, these have been deployed now in some clinical trials to see if they can be informative. Again, Dan Belsky did this nice paper looking at a rate of biologic aging. Again, his pace of aging measure, or his measure based on commonly measured blood chemistries and physiologic function. And he was able to show that in the calorie trial, where people were calorically restricted over time, that those undergoing caloric restriction had a slower and only slight increase in 2 years in their markers of their biologic age. So that's very promising. And most recently, this is also Dan Belsky's group. He presented this at GSA just last month, and it's in medRxiv Online. They looked at DNA methylation, and they looked at all these different clocks, and it was only the clock that he used with longitudinal measures in all of these blood chemistries that really distinguished between the calorically restricted and the ad libitum fed population. So this is telling us, I think, that lining up methylation to measures of biologic age based on things like chemistry, biochemistries, and looking at it over time has greater potential to be tracking the aging process than clocks that are just linked to chronologic age alone. So I'd just like to summarize. I hope I've convinced you that age is more than a number, and hope that when you're thinking about doing research in aging, that you consider looking at things like thresholds, age-specific subgroups, and potentially age as a exponential function in your analysis. Aging is modifiable, and because of that, we need intermediate biomarkers of age. The most sensitive measures to date have been those that are more closely related to diseases and risk factors for mortality than those that are only linked to chronologic age. And I expect that we'll see a continuing evolution in biomarkers that more closely reflect underlying biology. We're currently undertaking several studies using these cohorts like Health ABC and CHS, where we're looking at metabolomic profiles and proteomic profiles, which may help us with getting closer to the underlying biology of aging. So I'd like to close with this little cartoon from the New Yorker. The doctor says to his patient, "You're 57 years old. I'd like to get that down a bit." So maybe your doctor someday will have a biologic age readout to tell you whether there's potential targets to slow down or to better manage your aging process. With that, I'd like to close with an acknowledgement of the thousands of men and women in the epidemiologic cohort studies and clinical trials that I've worked with over the years with long-term NIH support, and the many collaborating investigators and postdocs at the University of Pittsburgh and nationally, as well as our Pitt Pepper Center and our Aging Institute at the University of Pittsburgh. So thank you very much for your attention, and I really enjoy participating in a discussion. Thank you so much.
Sorry. Again, thank you, Dr. Newman, for that wonderful talk, and for actually helping us think about the various, ways in which age can be, statistically at least thought through and rendered. So I have a question with regards to that. If we have multiple outcomes and, fairly limited power, what are some of the best ways to sort of think about, accounting for or taking the age component into consideration while statistically, analyzing the data and interpreting it? You did talk about the exponential, sort of way of including it in the analysis and potentially other ways, but are there some thoughts that you have? Because as you are aware, it's commonly adjusted as a covariate, and that's clearly, you've convinced us that is not the best way to deal with age.
Well, I do agree that age needs to be considered in almost any analysis unless you're studying a single age group, because there is so much variability. That's the one thing about aging that I'm sure those of you who study older people, they are so heterogeneous, and within any age, they're very heterogeneous. I think the important thing is just to think about what it means when you do adjust for age, and not just take it out of consideration. And I'm afraid that we have in the past often just done that, set it aside as a non-modifiable risk factor when we should really be thinking about what it means. When power is limited, it's always best to look at things as continuous variables, and using a squared term in a statistical model could be more useful than stratifying into age groups. But ultimately, you'd end up probably stratifying into age groups to communicate something if you do see a significant quadratic term.
Great, thank you. Does anyone else have questions that they'd like to ask? Roger, did you have any?
Yes. So Anne, thank you so much. And I'm sorry I had to come in late because I was standing by the treadmill doing a VO2 max test, which you talked about in your presentation.
Yes, a very critical test.
And, you know, I always get sort of, how are we going to get to a point where we know which biomarkers and which combinations of biomarkers are really the best ones? I mean, is this going to be sort of like regression tree analysis where they compete against each other, or, you know, how is that kind of unfold? Or is it going to be sort of AI that sort of helps us really understand that? Because I'm always sort of, I guess, overwhelmed with the potential number of, you know, whether there's senescence proteins or other biomarkers of aging. I'm just overwhelmed by the multitude of variables that we could use.
Yes, well, thank you for that question. And I have recently been doing some work in metabolomics, and it's mind-boggling how much information that you can have. And there are two challenges. One is an analytic approach, but the second is the having the depth of knowledge to interpret things that are biologically important, but maybe thrown aside because of multiple comparisons issue. So I think that we have to come at it from both directions and do some sort of data reduction technique, be that principal components or factor analysis, or which is basically, you know, there's sort of types of machine learning where you're setting criteria for staying in the model or not staying in the model. And going back to the basic biology of the knockout mice and the intervention studies, feeding studies, and things like that, and trying to bring them together. And I think it's going to take groups of investigators. I know that more and more I am working with the analytic people and the biochemists. And what I try to bring from my background in epidemiology and also geriatric medicine is reminding people that that there's a lot of levels of organization biologically that you can't really skip in your thinking. For example, many of the proteomic and metabolomic markers are cleared by the kidney, and older people tend to have less kidney function. And is it really driving their aging, or is it because they already have lost their kidney function that, and if you just knew their kidney function, you would have all the information. So I like to show that figure, especially when I'm working with my doctoral students about understanding every level of biologic organization, and don't just think simplistically that, well, this predicts very well An example might be TMAO, which is been found in metabolomics to be a cardiovascular risk factor, but it's also a uremic toxin. So, you know, what is the biology there? Is it something that you want to target some other aspect of kidney function, or can you target directly the TMAO? So we're finding things, and I think it's incredibly complex, and it'll take all of us working together, I think, to figure out where the best priorities are to pursue interventions.
Great. Thank you very much, Dr. Newman. We look forward to more of the work, and this was a fantastic and clear presentation and has given us a lot of food for thought. Thank you.
It's nice to see you all.
Thanks, everyone.
Thank you very